All right, good morning. These are the notes for how to build a protein part 2. This is translation. Last time we did transcription, which was step 1. Now we're going into step 2, which is how proteins are actually made. You need your level of organization paper. We're on the molecule or molecular level. You need to have your Cornell notes written down the side. At the top here, we've got translation. And it's a process where ribosomes create a protein from the mRNA recipe. So just by itself, the mRNA recipe doesn't do anything for you. It has to be translated into a protein, which is what your body actually needs. Okay? So after mRNA has made a copy of the protein recipe it needs, which is the gene, it has to take it to a place for the protein to be created. Okay, so this is like if you have a recipe and you got a recipe from the internet and you printed it out because you can't take the internet into your kitchen because it's no Wi-Fi zone. You're going to create what was in that recipe. You can't just eat the paper. You have to translate the paper into a dish. Okay, so for your cell, that place is a ribosome. Once mRNA arrives at a ribosome, the ribosome moves down the line and it uses transfer RNA to match amino acids with their codon. Okay, so if you remember, amino acids are the monomer or the building blocks of proteins, and this is how proteins are made. Each amino acid is attached to one transfer RNA. So these little T-looking things here, those are a transfer RNA, and they have an amino acid attached to them. Okay, so a ribosome, every 3 letters is a codon. Okay, so a codon is a set of 3 bases that code for a specific amino acid. There are 20 amino acids altogether. Every 3 bases, so there's 1 amino acid, there's 1 amino acid, that's 1 amino acid, that's 1 amino acid, that's 1 amino acid, and it just goes down the line. There's actually a chart that will tell you what amino acids are what bases, codons. So this ribosome goes down the line reading the code, and every time it gets to a codon, it says, okay, well, this is CUA, so that's going to be leucine. So it's going to attract a leucine to it, and it's going to attach it to this chain, and this chain is going to come out the end. And then when it's done attaching leucine, it's going to move to the next 3. Okay, that's tyrosine. So it's going to get a tyrosine, and it's going to attach that. Okay, then it's going to go down to the next one. All right, that's a arginine, so it's going to attach an arginine. And it's going to end up with this long chain of amino acids. When it gets to the end, it has created the entire protein, which is just one long chain of amino acids. Okay, so an example of a codon for these amino acids is AUG, that's a tyrosine. GCC, that's an arginine. So these amino acids translate into these codons. Okay, so you need to draw this picture. You need to 100% color it the way that I have it colored. The amino acids are blue. The tRNA is red. You need to color all of the bases. And the ribosome, you need to label. This is the mRNA strand. This is the amino acid chain, which is what a protein is. tRNA and ribosome. So in biology, there's a thing called central dogma, and it just basically means this is why we are alive. This is the basis of life. This is what causes you to be who you are. And it goes in this order: DNA, RNA, protein, trait. So DNA is the recipe. RNA copies the recipe. Then it also makes the protein. And then when you have that protein made, that's a trait. Okay, and that's what's called central dogma. Okay, over here on the side, your notes. Okay, so there are 20 amino acids, and that's it. There's just the 20, and that's everything that you're made out of. mRNA, it means messenger RNA, and tRNA is transfer RNA. Okay, this is on page 62, and your foldable is going to be on page 61.